Hi, I'm Sania, and in this podcast, I'm going to take you on a tour of the cell through 5 sets of sequential activities. Before I start talking about the activities, I would like to talk about the concept map of the cell. Let's start the understanding of the concept map through the definition of a cell. A cell is the basic unit of structure and function in all living organisms. Cells are broadly classified into 2 groups, prokaryotic cell and eukaryotic cell. Prokaryotic cell includes bacteria, cyanobacteria and archaea, whereas eukaryotic cell includes animal, protist, plant and fungi. Prokaryotic cells are generally found in unicellular organisms, whereas eukaryotic cells are found in both multicellular and unicellular organisms. Cells can be viewed through microscope. Before starting activities, students have been divided into groups comprising of 5 individuals per group. The following materials have been provided for each group of students on a table at the center of the laboratory. A handout has been given for each group of students to instruct each of the 5 sets of activities. Activity 1 is basically the observation of different types of prokaryotic and eukaryotic cell structures in terms of size and shape in pond water sample. Activity 1 is divided into 2 parts. Part A is the wet mount slide preparation for pond water according to the procedure shown here. In Part B of Activity 1, students have learned how to use a digital light microscope followed by viewing the prepared wet mount slide of pond water. Activity 2 basically involves observation of structure of human cheek epithelial cells under the microscope. In this activity, wet mount slide of human cheek cells has been prepared according to the procedure illustrated here, followed by successful observation of different cell parts such as nucleus and cell membrane under the microscope. In Activity 3, wet mount slide of Ehrlichia plant cells has been prepared according to the procedure illustrated here, followed by successful observation of different parts such as chloroplast cell wall under the microscope. In Activity 4, we have observed that the small molecules such as water can move into and out of the Ehrlichia leaf cells through the process of osmosis. When water molecules get into the leaf cells, the cell body expands and retains its shape. Whereas, when water molecules get out of the leaf cell, the cell membrane shrinks and pulls away from the cell. Activity 5 is basically a discussion session based on a set of quizzes on conceptions and misconceptions about the concept cell. In this activity, the quiz handout has been given to each student to actively participate in discussion with their teacher and circle the correct answer. Justification. At first, I would like to justify aforementioned activities from conceptual and contextual learning point of view, which include concept map and sequential activities. At the beginning of the activities, a concept map of cell has been shown to students, which help students to simplify the concept of cell and understand the interlinkages between different types of prokaryotic and eukaryotic cells with multicellular and unicellular organisms. Different parts of prokaryotic and eukaryotic cells and their functions, as well as cell division and its link to cell theory. The overall learning model presented here to teach the concepts is basically group activity-based learning, where each activity has been chosen logically and sequentially with real-life situation examples in order to facilitate conceptual and contextual learning abilities of students. For example, pond water sample has been chosen in Activity 1 since it provided the opportunity to students to observe and compare different types of prokaryotic and eukaryotic cell structures in terms of size and shape on the same slide. In Activity 2 and 3, human cheek epithelial cells and Ehrlichia plant leaf cells have been chosen to further explore students with eukaryotic cells and to develop the concept that cells are grouped together and are organized into body structures. In Activity 4, the same Ehrlichia wet mount slide prepared in Activity 3 has been chosen. to make the activity sequential, well-linked, and easily understandable to students, and to develop students' concept that cell is not only the basic unit of structure, but also the basic unit of function. Secondly, I would like to justify the activities in terms of alternative conceptions or misconceptions. 43% 6 to 8 graders have the misconception that all cells are the same size and shape. Basically, there is a generic cell. But in Activity 1, students got an opportunity to compare different sizes and shapes of cells, such as bacterial cells, protozoal cells, cyanobacterial cells, and other animal cells. 39% 6 to 8 graders have the misconception that there are no single-celled organisms. But in Activity 1, students have been seen single-celled organisms like bacteria, protozoa in pond water under the microscope. 39% 6 to 8 graders have the misconception that cells are not organized into body structures of the organism they are part of. But in Activity 2 and 3, students have seen their own chick cells are organized into body structures. Students have also seen that Elodea leaf cells are organized into body structures of Elodea plant. Many Stage 4 students have the misconception that cells are not the basic unit of function. They tend to think of cells as the stuff that makes up living organisms. But in Activity 4, students got an opportunity to see one of the major functions of a cell, that is, the transport of water molecules into and out of the cell through the process of osmosis. Many Stage 4 students have the misconception that everything a cell needs gets into it by diffusion. But in Activity 4, students have observed the diffusion of only water molecules into and out of the cell through the process of osmosis. Thirdly, I would like to justify the aforementioned activities in terms of activity-based learning. As mentioned earlier, 5 sequential sets of activities are involved in this learning process. Activity 1 to 4 all provided hands-on experiences to students in terms of microscopic slide preparation, use of microscope, observing osmosis and plasmosis in Elodea leaf cells. Additionally, Activity 1 to 4 provided cooperative learning among students via group activity. The learning techniques applied in Activity 5 also foster active learning through active participation of students via discussion with their teacher as well as through quiz tests. Fourthly, I would like to justify the aforementioned activities from teaching or learning point of view. 5E model has been successfully employed in the aforementioned learning activities. Engagement of students was achieved through the use of well-instructed, colorfully illustrated, and easily understandable procedures, as well as through the use of concept map to establish link with students' preconception. Exploration was achieved through the group activity. 5 students working in a group shared and explored their ideas through hands-on experiences, providing in each set of the aforementioned activities. Elaboration was achieved when the set of activities students performed challenged them to apply to a new situation surrounding them, such as water quality, testing, kitchen osmosis, and diagnostic test. Explanation and evaluation were achieved together through teacher-student discussion and quiz test on misconceptions and conceptions, where teacher clarified students' understanding of concepts through discussion and evaluation and helped them to develop their learning skills. Thank you for watching the Journey to Cell.